Welcome back to Summarize Science, where we break down complex research into simple terms. Imagine the inside of a living cell, not as a calm bag of water, but as a bustling city, full of tiny machines constantly at work. These machines, called enzymes, perform chemical reactions that are essential for life. This paper investigates a wild idea. Can the physical motion of these enzyme machines, separate from their chemical work, actually help organize the cell? To test this, the researchers focused on two key players. The first is a protein called ubiquinol, which, under the right conditions, clumps together to form little liquid droplets, a process called phase separation. The second is an enzyme called urease, which acts as their tiny motor. When urease is fed its fuel, urea, it becomes highly active and starts moving around much more rapidly, creating what the scientists call an active bath. A major challenge was to make sure they were only observing the effects of the enzyme's physical motion, not the chemical exhaust it produces. To do this, they built a special microfluidic chamber with semi-permeable walls. This clever device continuously supplied the enzyme with fresh fuel while simultaneously washing away the chemical byproducts. So, what happened when they turned on the enzyme motors in the presence of the phase-separating protein? The results were clear and dramatic. The active bath caused the protein droplets to grow significantly larger compared to the controls. Not only that, but the density of the droplets increased, meaning more droplets formed in the same amount of space. Essentially, the enzyme activity was driving more of the protein to condense into these droplets. The researchers also found that the concentration of protein inside each droplet was much higher in the active bath. This confirms that the physical jostling from the enzymes was actively pushing more protein molecules into the condensed, droplet state. To understand why this was happening, they ran computer simulations. The simulations showed that the larger, active bath particles effectively corralled the smaller protein polymers, almost like shepherds herding a flock. This herding action pushed the proteins closer together, enhancing their interactions and promoting the growth of larger clusters. The activity was essentially acting like an effective temperature to remodel the material. So let's recap the big picture here. This study provides the first direct demonstration that the physical activity of enzymes can influence protein organization at the nanoscale. It's not just about the chemical reactions enzymes perform, their very movement is a powerful force for order. This introduces a new physical principle for how our cells might manage their incredibly crowded internal environment. Inside the chaotic soup of a cell, the constant motion of countless enzymes could be a fundamental mechanism that helps create structure. Looking to the future, understanding these principles could allow scientists to design new active materials that assemble themselves in controlled ways. Thanks for watching Summarize Science.